Confused by how nucleotides actually build DNA and RNA, and why the MCAT keeps testing those tiny details? If terms like phosphodiester bonds, base pairing, and strand directionality feel abstract, you're not alone. In this video, we'll break nucleic acids down into simple, high-yield pieces so you can quickly recognize structures, predict bonding, and connect it all to function. By the end, you'll see DNA and RNA less as memorization and more as a system you can reason through on test day. Nucleotides are composed of a nitrogenous base, a pentose sugar, and one or more phosphate groups, forming the fundamental building blocks of nucleic acids. Think of each nucleotide as a three-part unit. The base provides identity, the sugar forms the backbone, and the phosphate enables polymerization. The nitrogenous bases are divided into purines, which are double-ringed adenine and guanine, and pyrimidines, which are single-ringed cytosine, thymine, and uracil. A common mnemonic is pure as gold for purines. The sugar is either ribose in RNA or deoxyribose in DNA, and the key difference is the 2-hydroxyl group present in ribose but absent in deoxyribose, which makes DNA more chemically stable. The base attaches to the 1 carbon of the sugar via an N-glycosidic bond, while the phosphate attaches to the 5 carbon. When only base plus sugar is present, it is called a nucleoside, and adding phosphate converts it into a nucleotide, a distinction that often appears in MCAT questions. Functionally, nucleotides are not just structural units. ATP is a nucleotide that serves as the cell's energy currency, and cyclic AMP acts as a signaling molecule, connecting this topic to metabolism and cell signaling. On the MCAT, you may be asked to identify components, recognize differences between nucleosides and nucleotides, or predict how structural changes, like loss of the 2-OH, affect stability and reactivity. DNA and RNA differ in sugar composition, nitrogenous bases, and structural organization, leading to distinct biological functions. DNA contains deoxyribose and uses thymine, whereas RNA contains ribose and uses uracil in place of thymine. Remembering DNA has T, RNA has U is high yield. The absence of the 2-hydroxyl group in DNA makes it less reactive and more stable, which is why it is ideal for long-term genetic storage, while RNA's 2-OH makes it more prone to hydrolysis and better suited for transient roles like messenger RNA, transfer RNA, and ribosomal RNA. Structurally, DNA is typically double-stranded, forming the classic double helix, while RNA is usually single-stranded and can fold into complex secondary structures, like hairpins. These structural differences directly relate to function. DNA stores genetic information, while RNA is involved in gene expression and catalysis, as seen in ribozymes. On the MCAT, you may encounter questions that test your ability to distinguish DNA from RNA based on chemical structure, predict stability differences, or interpret experimental data such as susceptibility to alkaline hydrolysis. This concept also connects to transcription and translation, where DNA is transcribed into RNA, reinforcing the central dogma of molecular biology. Phosphodiester bonds link nucleotides together to form nucleic acid polymers with a directional 5-3 backbone. Specifically, the phosphate group attached to the 5 carbon of one nucleotide forms an ester linkage with the hydroxyl group on the 3 carbon of the adjacent nucleotide, creating a repeating sugar-phosphate backbone. This linkage gives nucleic acids directionality, meaning one end has a free 5-phosphate and the other has a free 3-hydroxyl group, which is crucial for processes like DNA replication and transcription that proceed in the 5-3 direction. A helpful way to remember this is that new nucleotides are always added to the 3 end, because DNA polymerase requires a free 3-OH group. The phosphodiester bond is a covalent bond, making the backbone strong and stable, while the bases extend outward to participate in hydrogen bonding. On the MCAT, questions often test your understanding of directionality, asking which end of a strand is being extended or how antiparallel strands align. You may also be asked to identify where cleavage occurs during hydrolysis or enzyme activity. This concept connects to enzyme function, especially polymerases and nucleases, and to energy usage, as forming these bonds involves nucleotide triphosphates, like ATP or DNTPs. Complementary base pairing through hydrogen bonds stabilizes the DNA double helix and ensures accurate replication. In DNA, adenine pairs with thymine via 2 hydrogen bonds, while guanine pairs with cytosine via 3 hydrogen bonds, making GC pairs slightly stronger and more thermally stable. A common mnemonic is AT has 2, GC has 3. These specific pairings arise from the chemical structures of the bases, ensuring that purines always pair with pyrimidines to maintain a consistent helix width. The 2 strands run anti-parallel, meaning one runs 5 to 3 and the other runs 3 to 5, which is essential for proper alignment and replication. Hydrogen bonds are individually weak but collectively strong, allowing the strands to separate during replication and transcription without breaking the covalent backbone. On the MCAT, you may be asked to calculate melting temperature, predict the effect of base composition on stability, or identify mismatches. This concept also connects to mutations, where incorrect base pairing can lead to substitutions, and to techniques like PCR, where temperature changes disrupt and reform hydrogen bonds to amplify DNA. Nucleic acids store, transmit, and express genetic information through DNA replication, transcription, and translation. DNA serves as the long-term repository of genetic information, organized into sequences of nucleotides that encode proteins. During replication, DNA is copied semi-conservatively, meaning each daughter molecule contains one original strand and one newly synthesized strand, a concept frequently tested on the MCAT. Transcription converts DNA into messenger RNA, which carries the genetic code from the nucleus to the ribosome, where translation occurs and amino acids are assembled into proteins based on codon sequences. Each codon consists of 3 nucleotides, and this triplet code determines the amino acid sequence, linking nucleic acids directly to protein structure and function. Transfer RNA acts as an adapter, and ribosomal RNA forms the catalytic core of the ribosome, highlighting the diverse roles of RNA. On the MCAT, you may need to interpret mutations, predict how changes in nucleotide sequence affect protein function, or understand regulatory mechanisms. This concept integrates with many other topics, including enzyme kinetics, gene regulation, and biotechnology techniques like PCR and sequencing, making it one of the highest yield areas in biomolecules.